Hello everybody, this is Ola. In this video, I'll discuss some introduction about our neurotransmitters, synapses, and receptors. Remember that when the action potential goes along the axon and goes down to the axon terminals, this opens voltage-gated calcium channels. So calcium enters inside the axon terminals, and when they enter, they kick out the neurotransmitter here into the synapse. Then the neurotransmitter interacts with its own receptors and it opens up ligand-gated channels at the dendrites and the cell body of the postsynaptic cell. This would activate the postsynaptic cell and the neurotransmitter, and so the synapse can be either excitatory or inhibitory. Excitatory if a net influx of positive ions enter the cell, so the membrane goes positive, and here action potential is likely to happen if you go up to the threshold. Also, the synapses or the neurotransmitters can be inhibitory if a net influx of negative ions go into the cell, so they gain negative ions or positive ions exit the cell. So this is hyperpolarizing neurotransmitter, and this is inhibitory because it takes the membrane away from the threshold, so action potential is unlikely to happen. So what is the difference between excitatory and inhibitory synapses? The excitatory ones, they receive a depolarizing graded potential. The inhibitory ones receive a hyperpolarizing graded potential. And here for the excitatory ones, there is a net movement of positive ions into the cell. And in the inhibitory ones, either potassium, which is positive, exits the cell, or chloride ions, and they are negative, enter the cell, leading to hyperpolarization. In case of excitatory synapses, action potential is likely to happen, but it's not likely to happen in case of inhibitory synapses. An example of an excitatory neurotransmitter is epinephrine or norepi, glutamate, and substance P. An example of an inhibitory neurotransmitter is glycine, GABA, and endorphins. Now, the neurotransmitter is in the synapse doing its action on the postsynaptic cell. When it's done with its action, what will happen? The neurotransmitter must be removed from the synaptic cleft, because if it continues to be in the synapse, the continual stimulation of the postsynaptic cells can lead to injury of them. So we need to stop the stimulus by removing the neurotransmitter from the synapse. So how does the neurotransmitter leave the synapse? There are three ways. Either the neurotransmitter simply moves away from the synapse, or it can be broken down by an enzyme. And our example here is acetylcholine. When acetylcholine is done with its function stimulating the postsynaptic cell, it is going to be broken down by an enzyme which we call acetylcholinesterase enzyme, and then it can be recycled. So here we have our acetylcholine in the synapse. After it's done with its job on the postsynaptic cell, it must be removed or broken down by the acetylcholinesterase enzyme. So it's gonna just break down acetylcholine, and then it can be recycled to be used later. The third way is that the neurotransmitter can be taken back into the presynaptic cell. We call that reuptake. So here we have the neurotransmitter in the synapse. After doing its job, it's gonna go back to be stored again into the presynaptic cell through reuptake channels. If you look here to acetylcholinesterase enzyme that breaks down acetylcholine, there is something which is the organophosphate poisoning. This inhibits the acetylcholinesterase enzyme from doing its job, which is breaking down acetylcholine. So here is the organophosphate poison, and it blocks the acetylcholinesterase enzyme, preventing that from breaking down acetylcholine. So what would be the result of that? Acetylcholine will stay in the synapse, will continue stimulating its post-synaptic neuron, leading to different symptoms of organophosphate poisoning due to the excess acetylcholine. So let's go with these two review questions. Inhibitory neurotransmitters cause what in the postsynaptic neuron? Of course, they are inhibitory, so they will do hyperpolarization. So they are hyperpolarizing. On the other hand, excitatory neurotransmitters cause depolarization in the postsynaptic cell, so action potential is likely to happen.